Welcome to the deep dive, where we cut through the information overload and really unearth the essential insights from some fascinating sources.
Mm.
Today, we're embarking on a journey, really, into the very architecture of life. We're drawing our map from Lehninger Principles of Biochemistry, 8th edition, by Nelson and Cox, a true cornerstone of the field.
Right. Our mission essentially is to distill the core principles from its first chapter. And we're not just summarizing here, we're extracting the most profound nuggets, focusing on, well, the elegant molecular mechanisms,
Yeah.
the ingenuity of biochemical pathways, the logic of enzyme kinetics, the driving forces of thermodynamics, and that incredible dance of metabolic integration.
All presented to illuminate what truly matters for you, our curious listener. Think of it like an upper division undergraduate level, right?
Exactly. Accessible but deep.
Think of this as your expedited tour through the fundamental building blocks of biology. We're aiming for those genuine aha moments, revealing the astonishing fact that life, in all its breathtaking complexity, operates entirely within the same physical and chemical laws that govern the inanimate universe.
It's quite something. It's a story of matter organizing itself in, frankly, the most improbable ways.
So let's start at the beginning, the cell. Biochemistry ultimately asks, how do the incredible dynamic properties of living organisms emerge from what are, at their core, just collections of molecules?
That's the core mystery, isn't it? Consider this. Despite the dizzying diversity of life around us, from bacteria to blue whales,
Right.
every living organism shares fundamental defining features. They are all made of cells, the basic units of life.
Okay.
They constantly extract energy and maintain a dynamic, steady state far from equilibrium, and precisely self-replicate using genetic information. And critically, they all evolve over time.
It's just astounding that such complex functions arise from such small packages.
Huh.
What's universally true for almost all cells, then?
Well, they're enclosed by a plasma membrane, a flexible hydrophobic barrier made of lipids and proteins that acts as the ultimate bouncer, really.
Controlling what gets in and out.
Precisely. Inside, you've got the cytoplasm, which is this aqueous wonderland, the cytosol, packed with enzymes, RNA, amino acids, all the molecular players.
Got it.
And every single cell, at some point in its life, houses its genetic blueprint, either in a sort of nucleoid region, unbound by a membrane, like in bacteria and Archaea, or within a true nucleus, neatly packaged within a double membrane. That's the hallmark of eukaryotes like us.
You might wonder, though, what truly dictates a cell's size? I mean, the lower limit seems practical. You just need a minimum number of biomolecules to actually do anything, right?
That's right. But the upper limit is far more fascinating, I think. It's all about logistics, specifically the rate of nutrient and waste transport. Think about it this way. As a cell gets bigger, its volume increases much faster than its surface area.
Right, the cube versus the square thing.
Exactly. A spherical cell's surface area goes up with the square of its radius, but its volume explodes with the cube.
So small cells are just more efficient.
In that respect, yes. A tiny bacterial cell like E. coli has an enormous surface-to-volume ratio. That makes it incredibly efficient at absorbing nutrients and expelling waste.
But bigger cells exist, obviously.
You do.
Hmm.
And larger cells, like our own lymphocytes, often evolve intricate folds or, specialized transport systems to compensate, just ensuring every part of the cell gets what it needs. It's a fundamental constraint, really, that shapes cellular architecture.
Okay, here's where it gets particularly insightful for me. From a genetic and biochemical standpoint, all life on Earth branches into these three domains: bacteria, Archaea, and Eukarya. And a truly surprising finding from molecular studies, wasn't it?
Yeah.
That Archaea are actually more closely related to us, the Eukarya, than either is to bacteria.
Well, it's a big shift, yes. It definitely rewrote some of the traditional biological family trees. And we can also classify organisms by their metabolic strategies, basically, how they get energy and carbon.
Okay.
You have phototrophs, like plants and cyanobacteria. They harness sunlight. Then chemotrophs, like us, oxidize chemical fuels.
And then autotrophs and heterotrophs.
Right. Autotrophs synthesize their own biomolecules from simple CO2, while heterotrophs, like us again, need preformed organic nutrients.
So a cyanobacterium is a photoautotroph.
Exactly. Pulling energy from light and carbon from the air.
Well, you and I are classic chemoheterotrophs.
Relying on the organic compounds we consume.
Correct. Moving inside the eukaryotic cell, it just feels like a whole new level of organization. These cells are much larger, And they feature this whole variety of membrane-enclosed organelles, each like a specialized factory.
That's a good analogy. Mitochondria are the energy powerhouses. The endoplasmic reticulum and Golgi are all about protein and lipid processing. Lysosomes are the cell's recycling centers.
And it's not just a sack of organelles floating around, is it?
Not at all. The cytoplasm is intricately organized by the cytoskeleton, this dynamic meshwork of protein filaments like actin, microtubules, and intermediate filaments.
Dynamic is the key word there.
Absolutely. These aren't static scaffolding. They're constantly assembling and disassembling, giving the cell shape, organizing its internal components, and enabling motion. And this constant regulated dance is entirely driven by individually weak, non-covalent interactions.
Like hydrogen bonds.
Hydrogen bonds, hydrophobic effects, ionic interactions, yeah. Billions of tiny interactions working together.
So that leads to this fundamental principle. Life operates on a structural hierarchy. Small, monomeric subunits build into macromolecules.
Proteins, nucleic acids.
Right, which then spontaneously self-assemble into these large supramolecular complexes like ribosomes or membranes.
And it's not just random assembly, it's directed assembly, orchestrated by those same non-covalent interactions. It's quite elegant. This brings us to a crucial point, though, about how we study biochemistry. While we often isolate and study purified molecules in vitro, in a test tube,
Which simplifies things.
it does. But the cellular environment in vivo is anything but simple. It's an incredibly crowded, bustling metropolis of molecules.
Right.
What looks like simple diffusion in a nice, clean test tube is actually a chaotic obstacle course inside a cell. Understanding how this intricate cellular organization, this internal crowd, fundamentally influences molecular function is one of the central ongoing challenges in biochemistry today.
Okay, shifting gears slightly to the chemical side, one of the most profound aha moments for me in biochemistry is this idea of the biochemical unity of life.
Yes, Monod's quote.
Exactly, Jacques Monod. What is true of E. coli is true of the elephant. And that isn't just a clever saying, right? It means the fundamental chemical intermediates and transformations are astonishingly conserved across all organisms.
It's a powerful reflection of a common evolutionary origin, and at the heart of this unity is carbon,
The backbone.
the ultimate versatile backbone of life.
Yep.
It's capable of forming stable single and double bonds with itself, hydrogen, oxygen, nitrogen.
Which allows for huge diversity.
Enormous diversity of molecular shapes, linear chains, branch structures, rings forming the complex scaffold of biomolecules. No other element really offers such flexibility for building diverse structures.
So most biomolecules are basically these carbon skeletons with various functional groups attached, like hydroxyls, amino groups.
Precisely. Those groups are the chemical personalities, defining how a molecule interacts and reacts. Take acetyl coenzyme A, for example. It's a polyfunctional molecule just brimming with different groups, and each one contributes to its central role in metabolism.
And here's another unifying theme. Dissolved in the cytosol of pretty much every cell is this remarkably conserved universal set of small organic molecules, maybe a few thousand different kinds.
That's about right. These are the central metabolites, amino acids, nucleotides, sugars. They are polar or charged, water-soluble.
And crucially,
The plasma membrane traps them inside. This allows the cell to build and maintain its own unique chemical environment distinct from its surroundings.
beyond these universal building blocks, then we get the big guys, the macromolecules, proteins, nucleic acids, polysaccharides, lipids.
Right. Proteins, built from amino acids, are incredibly versatile enzymes, structures, signals, transporters. Nucleic acids, DNA and RNA, store and transmit genetic info. Polysaccharides store energy, provide structure. Lipids form membranes, store energy.
And proteins and nucleic acids are often called informational macromolecules.
Yes, because their linear sequence of subunits contains critical information that dictates their function. And this brings us straight to stereochemistry, the crucial 3D arrangement of atoms. This absolutely dictates how these molecules interact.
Okay, so let's unpack that. When we talk about configuration, we mean fixed spatial arrangements like geometric isomers, cis trends.
Exactly. Think of retinal in your eye. Light flips one isomer to another, and that tiny change triggers the entire cascade of vision.
Wow.
Or consider a chiral center, usually a carbon atom bonded to 4 different groups. This creates enantiomers.
Mirror images that aren't superimposable.
Precisely. Non-superimposable mirror images. Louis Pasteur famously separated the two enantiomers of tartaric acid,
Right, I remember that story.
showing they had identical chemical properties but dramatically different optical properties, how they rotated polarized light.
And this is incredibly vital in biology.
Oh, absolutely. For example, the amino acids found in proteins exist almost exclusively in one chiral form, the L-isomer. Our bodies are incredibly selective.
Which explains why biological systems are so discriminating. Like, take the artificial sweetener aspartame. It tastes sweet, but its stereoisomer, differing at only one chiral carbon, tastes bitter. Our taste receptors can tell them apart perfectly.
It's amazing specificity. Or think about the antidepressant citalopram. Only one of its enantiomers actually works therapeutically.
It's like trying to fit a left-handed glove onto a right hand.
Exactly. Biomolecular interactions are profoundly stereospecific. 3D shape is paramount to function.
Okay, so that's configuration, which is fixed. What about conformation?
Right. Distinct from configuration is conformation. This refers to spatial arrangements that can change by rotation around single bonds.
So more flexible.
Potentially. While simple molecules like ethane might have pretty free rotation, in complex biomolecules, bulky or charged groups often restrict this rotation. This leads to specific stable conformations or shapes. And these preferred shapes are absolutely crucial for a protein to do its job, for an enzyme to bind its substrate, or for DNA to form its double helix. It's all about the right shape.
Okay, now for one of life's, maybe the central mystery, how do living cells perform the work necessary to stay alive, grow, reproduce? How do they harness energy in a universe that, constantly trends toward disorder?
That's the big question. Living organisms are classic open systems. They're constantly exchanging both matter and energy with their surroundings.
Not closed up.
Definitely not. And they exist in a dynamic, steady state. That means their composition stays relatively constant, but the molecules inside are in a state of furious, continuous flux, being synthesized, broken down, replaced.
So the hemoglobin in my blood isn't the same hemoglobin I had last month.
Not at all. It's constantly being replaced. This steady state is maintained far from equilibrium, and that demands a constant, enormous investment of energy.
And if the energy stops?
The moment a cell can't get energy, it dies and decays. It moves irreversibly toward equilibrium, which for a cell means death and dissolution.
Which brings us right to the laws of thermodynamics. The first law, energy, is conserved, right?
Correct. It can change forms, but the total amount in the universe is constant. Cells are absolute masters of energy transduction, converting chemical energy into mechanical work, electrical signals, osmotic gradients.
Efficient conversion, not creation.
Precisely. But the second law is arguably even more fascinating for biology. It states that the universe tends toward ever greater disorder, or entropy.
So how does life, this highly ordered system, even exist?
Great question. To create and maintain the incredibly complex and ordered state of a living cell, work and energy are absolutely required to counteract that natural tendency towards entropy.
So life exports disorder.
In a way, yes. When glucose is oxidized, for instance, 7 molecules of reactants turn into 12 molecules of product. That increases molecular disorder in the surroundings, releasing free energy. This release of energy and increase in entropy outside the cell effectively pays for maintaining the order inside the cell.
Okay, so where does the energy for this constant battle against entropy actually come from?
Almost all life on Earth relies on the flow of electrons, fundamentally. Electrons moving from a higher energy state to a lower one.
How does that work?
Well, photoautotrophs capture sunlight to split water, releasing high-energy electrons and oxygen. Chemotrophs, like us, get energy by oxidizing the energy-rich products of photosynthesis, ultimately passing electrons down an energy gradient to oxygen.
Like a tiny biological electrical circuit.
That's a very good analogy. It's like a controlled downhill cascade of electrons releasing energy in manageable steps.
Which leads us to free energy.
Exactly. A process, a reaction, will occur spontaneously only if the change in free energy is negative. That means free energy is released. These are exergonic reactions.
And if DCE is positive?
The reaction is endergonic. It requires an input of energy to proceed. It won't happen on its own.
And here's where the magic in biology seems to happen. Energy coupling.
It does seem magical, but it's pure chemistry. An exergonic reaction, one that releases energy, can be directly coupled to an endergonic one.
Driving the unfavorable one forward.
Precisely. Think of it like a large, heavy object falling spontaneously, and you use a pulley system to lift a smaller object uphill.
Okay.
In cells, the breakdown of ATP adenosine triphosphate is a hugely exergonic reaction. It's the primary energy currency.
So ATP breakdown pays for other reactions.
It does. The hydrolysis of ATP to ADP and inorganic phosphate releases a large amount of free energy. Critically, this isn't just because of its chemical bonds, but because the cell works hard to maintain ATP's concentration far above its equilibrium concentration.
So this is a chemical tension.
A powerful chemical tension, yes, waiting to be released. This disequilibrium allows ATP to serve as the major carrier of chemical energy, driving countless endergonic reactions.
Like making glucose 6-phosphate.
That's a classic example. Making glucose 6-phosphate from glucose and phosphate is energetically unfavorable on its own. But couple it with ATP hydrolysis, and the overall reaction becomes favorable because the standard free energy changes just add up. It's an ingenious way life overcomes thermodynamic barriers.
Okay. But even if a reaction is thermodynamically favorable, has a negative barrier, why don't complex biomolecules just spontaneously fall apart all the time? Or why aren't reactions too slow to sustain life?
Ah, excellent point. This is precisely where enzymes come in. They are nature's incredibly elegant biocatalysts.
Speeding things up.
Massively speeding things up. They enhance the rate of specific chemical reactions, often by a millionfold or even much more, without being consumed in the process.
How do they do that?
They work by lowering the activation energy. That's the energy barrier reactants must overcome to reach a high-energy transition state before they can form products.
So they make the hill smaller.
Effectively, yes. Enzymes provide a unique chemical environment, a binding site, that perfectly complements and stabilizes this transition state. This dramatically increases the reaction rate, making life's rapid, complex chemistry possible at body temperature.
And these thousands of enzyme-catalyzed reactions, they're not random, are they? They're organized.
Highly organized into interconnected pathways. This network of pathways is what we call metabolism.
And we usually divide that into catabolism and enablism.
That's the common division. Catabolism involves degrading nutrients, breaking things down to extract energy and produce ATP. Enablism is the opposite, building complex molecules from simpler precursors, which requires energy input, usually from ATP.
So ATP is the link.
ATP, along with electron carriers like NADPH, acts as the central link, the kind of energy bridge between catabolic energy release and anabolic energy consumption. It's truly the cell's universal currency, and this whole intricate metabolic network is under incredibly tight regulation, often through mechanisms like feedback inhibition, where the end product of a pathway inhibits an enzyme early in the pathway.
So the cell doesn't make too much of something.
Exactly. It ensures the cell produces exactly what it needs when it needs it, and does so with remarkable efficiency, preventing waste. That's metabolic integration in action. It's beautifully economical.
Okay, let's turn our attention now to maybe one of the most astonishing properties of life. Its uncanny ability to reproduce itself with nearly perfect fidelity.
Across
Countless generations.
It is astonishing.
The genetic instructions for building an organism have remained, well, astonishingly constant for billions of years. How is that even possible?
Think about this scale. The single DNA molecule of an E. coli cell has about 4.6 million nucleotide pairs.
That's a lot of information.
It is. Now imagine hand copying a book with, say, 5 million characters, passing it on, and that copy being copied again trillions of times over 3.5 billion years, and each copy remains virtually identical.
That sounds impossible.
But that's essentially the mind-boggling fidelity of DNA replication.
So the secret must lie in DNA structure, that double helix.
Absolutely. It's a long linear polymer of 4 types of deoxyribonucleotides arranged in a precise sequence. The 2 strands twist around each other, but the real genius is in the specific pairing.
A with T, G with C, base pairing rules.
Adenine always with thymine, guanine always with cytosine. This complementarity is the absolute key to genetic continuity.
How so?
Before a cell divides, the 2 DNA strands simply separate, unzip, and each one serves as a perfect template for synthesizing a new complementary strand.
So you get 2 identical copies from one original.
Exactly. 2 identical double helical molecules maintaining the genetic information with incredible accuracy. And there's a bonus. If one strand gets damaged.
Which must happen sometimes.
Oh, it does. If one strand is damaged, the other intact strand provides the necessary information for a high...
Okay, so we have this one-dimensional sequence of nucleotides in DNA. A's, T's, G's, C's. But that somehow dictates the formation of a complex 3-dimensional cell. How does that leap happen?
It's generally a two-phase process, often called the central dogma of molecular biology. DNA codes for an intermediary molecule, RNA,
Right. Transcription.
which then codes for a linear sequence of amino acids in a protein.
Translation.
Exactly. DNA to RNA to protein.
But a linear sequence of amino acids is still just a chain. How does it become a functional 3D machine?
And here's another astonishing step. That linear protein chain then spontaneously folds itself into a precise three-dimensional shape, its native conformation.
Spontaneously, just based on the sequence.
Primarily guided by its amino acid sequence itself, yes. And stabilized by those familiar non-covalent interactions, hydrophobic effects driving non-polar residues inside, hydrogen bonds, ionic interactions forming.
And this specific 3D structure is absolutely crucial for function.
Utterly crucial. Think of an enzyme like hexokinase. It needs its perfect, intricate shape to recognize glucose and catalyze its phosphorylation. Shape determines function.
In these perfectly folded proteins, they don't just work alone, do they?
Often not. They can then spontaneously self-assemble with other macromolecules, other proteins, RNA, lipids, into larger supramolecular complexes.
Like ribosomes or chromosomes.
Exactly, like chromosomes, ribosomes, membranes. Each complex has specific high-affinity binding sites, ensuring accurate assembly, like intricate molecular Legos.
And the environment matters too.
The cellular environment, the right pH, ionic strength, presence of specific metal ions also plays a crucial role in ensuring this accurate folding and assembly. It really highlights the interconnectedness of all these foundational principles we've been discussing.
Okay, here's where it feels like it all truly comes together, revealing the grand sweep of billions of years, as Theodosius Dobzhansky famously said.
Nothing in biology makes sense except in the light of evolution.
Exactly. The remarkable similarities we see in metabolic pathways, in gene sequences, even in the basic molecular building blocks across all life forms, it strongly suggests a common evolutionary ancestor, doesn't it?
Overwhelmingly so. Evolution itself relies on genetic mutations, those infrequent, unrepaired mistakes in DNA replication that change nucleotide sequences.
The stakes are key.
Well, while many mutations are harmful or just neutral, occasionally one might serendipitously give an organism a selective advantage in its specific environment.
Allowing it to survive better, reproduce more.
Precisely. Leading it to survive and prosper through natural selection, what Herbert Spencer later called survival of the fittest. It's a relentless but blind sculptor of life over vast timescales.
You mentioned in the notes something really fascinating about how new functions arise, gene duplication.
Yes, that's a key mechanism. Imagine a gene is accidentally copied twice during replication. Now, one copy is essentially redundant. It's a spare.
So the original can keep doing its job.
Exactly. Mutations can then accumulate in this extra copy without immediately harming the organism. This allows the duplicate gene the freedom to potentially evolve a completely new advantageous function over time. It's a major way life innovates and builds complexity upon existing structures.
So going way back, the very first living cell, billions of years ago, must have faced this fundamental challenge. Where did it get its organic building blocks?
That's a huge question in origins of life research. One leading hypothesis is chemical evolution, the idea that biomolecules were produced abiotically, non-biologically,
From simple gases.
from simple gases like ammonia, methane, water, hydrogen, perhaps nitrogen, driven by powerful environmental energy sources like lightning or UV radiation.
The Miller-Urey experiment.
The classic Miller-Urey experiment in 1953 vividly demonstrated this possibility. Electrical sparks, simulating lightning, went through a mixture of simple gases.
They got amino acids.
They did, amino acids and other organic compounds. And remarkably, more recent analyses of Miller's original preserved samples, even vials containing things like hydrogen sulfide, have confirmed the formation of 23 different amino acids.
Wow. And hydrothermal vents.
Deep-sea hydrothermal vents, those black smokers, are another really strong candidate for the crucible of early biogenesis. They provide both raw chemical materials and geothermal energy gradients.
This leads into the RNA world hypothesis, right?
It does. It's a very compelling idea. It proposes that RNA molecules came first. RNA is unique because it can both store genetic information, like DNA, and act as a catalyst, like protein enzymes. These are called ribozymes.
So RNA was doing both jobs initially.
That's the hypothesis. This self-replicating RNA could then have evolved the ability to catalyze peptide formation, eventually leading to the specialization we see today. DNA becoming the primary, more stable information store, and proteins becoming the incredibly versatile catalysts. It's a plausible evolutionary pathway from a primordial soup to complex cellular life.
So life on Earth began over 3.5 billion years ago in an oxygen-deprived, reducing atmosphere. Early cells probably used inorganic fuels.
Most likely, yes. Then came a monumental shift. The evolution of photosynthetic organisms, particularly cyanobacteria.
The ones that use water.
Yes, they started using water as an electron donor for photosynthesis, and the byproduct was oxygen. This gradually, over millions of years, transformed Earth's atmosphere.
Leading to the oxygen holocaust.
For many existing anaerobic life forms, yes, oxygen was toxic. This dramatic change created immense evolutionary pressure.
But some adapted.
Some adapted brilliantly. This gave rise to aerobes, organisms that could not only tolerate oxygen, but could actually harness its strong electron-accepting power for respiration.
Getting much more energy out of their fuel.
Vastly more energy. This incredible energetic advantage led to their predominance in the newly oxygen-rich environments, fundamentally reshaping the entire biosphere.
What about the evolution of eukaryotic cells? Our kind of cells.
That happened later, maybe around 1.5 billion years ago. It involves several major changes. First, the evolution of the chromosome structure allowed for compacting and reliably dividing significantly larger amounts of DNA.
Okay.
Second, the nucleus evolved that membrane-bound compartment, elegantly segregating DNA replication and transcription from the bustling cytoplasm.
And third, endosymbiosis.
Third, and perhaps most profoundly, was endosymbiosis. Early eukaryotic precursor cells engulfed aerobic bacteria, and in some lineages, photosynthetic cyanobacteria.
And they stuck around.
They stuck around and formed a symbiotic relationship. These engulfed organisms eventually became mitochondria, the powerhouses of the cell, and chloroplasts, the sites of photosynthesis, respectively. This was a complete game changer, integrating powerful new metabolic capabilities directly into the host cell.
And eventually leading to multicellularity.
Yes. Later, single-celled eukaryotic organisms began clustering, cooperating, leading to true multicellularity and cellular differentiation, where different cells specialize for different tasks to support the entire complex organism, like in our bodies.
The power of modern science now, with molecular anatomy. We can sequence entire genomes.
It's incredible. We can reveal detailed evolutionary relationships just by comparing gene or protein sequences. Genes that are homologous, meaning they share readily detectable sequence similarities because they derive from a common ancestral gene, give us a direct molecular record of evolution. We can literally trace common ancestry and divergence in the DNA.
And this leads to things like functional genomics.
Exactly. Functional genomics allows scientists to look at the whole genome and see what proportion of it is dedicated to specific cellular processes. For example, you find that in more complex organisms, a larger chunk of the genome encodes genes involved in regulating cellular processes, rather than just basic housekeeping functions like making ATP.
That speaks to the evolution of control and complexity.
It really does.
In this knowledge, it obviously has truly incredible implications for medicine today.
Oh, absolutely. Large-scale genomic studies are identifying specific gene variants or mutations that directly correlate with susceptibility to complex diseases like cancer, heart disease, or diabetes.
So we're moving towards a future where maybe medical history gets augmented by a medical forecast.
That's the direction, certainly. Predicting disease risk based on our individual genetic makeup and potentially intervening preventatively. It's a profound shift in how we think about and approach health and disease.
Wow, what an incredible journey we've taken from the very atoms that make up our bodies, formed in stars, to the intricate dance of molecules within just a single cell, and then pulling back to see the grand sweep of billions of years of evolution. Biochemistry really does offer this unified, elegant view of life.
It really does. It highlights that despite the immense, almost bewildering diversity of life forms we see around us, the underlying molecular structures, the mechanisms, the metabolic pathways, they are remarkably fundamentally similar.
Governed by the laws of chemistry and physics.
Exactly. These fundamental principles, governed by the immutable laws of chemistry and physics, have just been relentlessly refined over eons of natural selection, culminating in the complex life we observe today.
And for you, our listener, hopefully the real beauty of this deep dive isn't just in, memorizing these fascinating facts, but in realizing the profound elegance of how seemingly inanimate matter can self-organize, sustain itself, and perpetuate life. Which kind of raises an intriguing question for you to ponder. What other fundamental chemical or physical laws might be shaping life, perhaps in ways we haven't even fully begun to discover yet?
A question for the future of science.
Indeed. Thank you for being part of the Last Minute Lecture family and for diving deep with us today.